All right. Our last chapter of the term deals with pregnancy and human development. And pregnancy is simply the series of events that takes place from implantation of the blastocyst until the birth of the neonate. And this is going to take approximately 9 months. The conceptus is defined as the developing offspring. The gestation period is the time from the last menstrual period until birth, which is approximately 280 days. The embryo is the conceptus from fertilization through week 8, and the fetus is from week 9 through birth and is marked by the appearance in week 9 of organs for the first time. You can see the difference in relative size in the diagram here. There are three processes that take place in order to turn something that's about the size of a head of a pin into a full-blown neonate. You start out with a single cell that results from fertilization, where the sperm penetrates the egg cell membrane and generates a zygote, the first cell in a new individual. After that point, three processes that take place in order to promote the development of the embryo are cell division, morphogenetic movement, where populations of cells move from one location to another, growth, where cells increase in size, and finally differentiation, where cells using chemical cues in the environment begin to change their behavior and their appearance, and this is driven by limiting the regions of DNA that those cells can use so that eventually they're restricted to only the genetic material that's going to define their final function in the adult. Okay? So, the oocyte, once it's out of the ovary, is viable for 12 to 24 hours, while the sperm are viable for 1 to 2 days following ejaculation. For fertilization to occur, intercourse has to occur no more than 2 days before or a day after ovulation. Fertilization is where the sperm's chromosome combine with those of the secondary oocyte and generate the first diploid cell of a new individual, which we define as a zygote. Okay, and an important point about this is that this cell is going to contain a completely unique distribution of genes that is different from either the mother or the father. So while the offspring will be similar to the mom and dad, they will not be identical. And this is the advantage of a sexually reproducing species, is the presence of extensive biodiversity, okay, meaning physical, physiological, and biochemical differences in the population upon which natural selection can act in order to pick out those characteristics that make it more likely that the organism will survive in that environment. Okay? Ejaculated sperm leak out of the vagina immediately after they're deposited and are destroyed by the vaginal environment and don't make it through the cervix. The remainder are dispersed in the uterine cavity or destroyed by phagocytes, and only a few reach the fallopian tubes. Okay, so the point in bringing this up is that you have to have a certain minimal sperm count in order to realistically accomplish fertilization naturally. And if you're below that, there's not a realistic chance of you fertilizing the egg because of all of these barriers. Okay, swimming up the wrong fallopian tube, being destroyed by the woman's immune system, attempting to fertilize the uterus or the cervix or the vagina. Okay, and then either overshooting or undershooting the egg if you do get into the correct fallopian tube. Okay, all of these are obstacles to fertilization. And then once you latch onto the egg, there's a few dozen sperm that use their acrosomal enzymes to penetrate first the corona radiata, which are the outer follicle cells that protect the egg from the woman's own immune system, and then the zona pellucida, which is an acellular matrix that also protects the egg from the immune system. And only one sperm is going to make it through the zona into the egg cell membrane, bind to the sperm cell receptors, and then be taken in to the egg and accomplish the fertilization. And at that point, the other sperm cells are locked out, okay? But they do stay attached to the outer layer of cells, the granuloses, in the corona, and they help paddle the egg towards the uterus. The sperm have to be motile in order for them to accomplish this, and so they get capacitated before they penetrate the oocyte. This enhances their motility, and it also causes the acrosomal membrane to become fragile enough so that when it hits the corona, the enzymes are released. The secretions of the female tract are designed to do that. The sperm follow a chemical trail in order to reach the oocyte, okay? So there is some help that the egg provides. The sperm have to breach the corona and the zona, okay? It weaves its way through the corona, binds to the zona, and at this point the acrosomal reaction happens where enzymes are released and digest holes in the zona. The net effect of these acrosomes release enzymes to digest the entire zona, and then a single sperm head will approach the oocyte. The back portion of the acrosome will bind to the oocyte plasma membrane. The oocyte and the sperm membranes fuse. The gametes fuse as the sperm's cytoplasmic contents enter the oocyte. And the rest of the sperm are locked out because of the fast and slow block to polyspermy. Okay? If more than one sperm were to penetrate the egg, the resulting cell wouldn't be a viable fertilization product, and you would end up with spontaneous abortion. And this probably does happen. It's just that it never gets recorded as a statistic because at that point the fertilization product passes through the reproductive system and we never know it was there. Okay? Once the first sperm head enters the egg by binding to the sperm receptor on the egg cell membrane, there's a calcium surge from the smooth ER. The cortical reaction takes place. Cortical granules release enzymes in exocytosis that contain zonal inhibiting proteins. The zips strip off the sperm receptors. Fluid binds water and swells and detaches the other sperm so that they're locked in the zona, but the zona is no longer in contact with the egg cell membrane. And you can see here the process that takes place, right? There's the corona, right, out here. These are the sperm binding and digesting, okay? The zona contains the sperm binding receptors, right? One of the sperms then penetrates, the cortical granules then release, right? And then we destroy those sperm cell receptors, but the sperm are still latched into the zona, and now they help paddle the egg towards the uterus, okay? At that point, the egg will quickly complete meiosis II and generate a second polar body. The sperm genetic material will swell to form the male pronucleus. A calcium surge triggers the completion of meiosis II in the second polar body. The female pronucleus swells, and then fertilization is the moment when the membranes of the pronuclei fuse and the chromosomes combine, okay, and that membrane vanishes, and then we undergo our first cell division. So you can see here the process, right? What we've got here is the corona. We've got the sperm nucleus in there, okay? We quickly complete the second meiotic division because we've been locked in metaphase meiosis II at this point. The second polar body forms, okay? And then you've got the male and female pronucleus. They fuse, the membranes dissolve, the spindle apparatus appears, and now we truly have the zygote, okay? The process whereby the male and female pronucleus fuse is called amphimixis, okay? And you can see here male and female pronuclei fusing, and the polar bodies on the periphery. This is an actual electron micrograph. Next thing to occur is cell division. We call this cleavage. This happens while the zygote is moving towards the uterus. Mitotic divisions take place. The first cleavage is at 36 hours, generating 2 daughter cells. At 72 hours, we have a cell mass called a morula, which is approximately 16 cells large. And at day 5, the blastocyst contains an embryo of approximately 100 cells that will reach the uterus. Now, the blastocyst is interesting because it's a combination of an extracellular layer, which forms a barrier around fluid, inside of which is an inner cell mass latched onto one side of the outer blastocyst cells. Okay, the inner cell mass is what's eventually going to become the embryo and the extraembryonic membranes, and the cells on the outside are going to form what we call the trophoblast. Okay, but this is basically a hollow ball of cells that will ultimately enter the fundus of the uterus and float there for about 3 days and then attach to the endometrium. Okay, so at this stage, we're a lot like a jellyfish. Okay, it's sort of a very primitive body structure here. The fluid-filled space is homologous to a hydroskeleton. Okay, and animals with hydroskeletons include starfish and jellyfish. Right, these are fluid-filled inner chambers that give the organism shape. And this is one of 3 events in the development of what will eventually be the skeletal system of the organism. Okay, so the trophoblast cells on the exterior have immunosuppressive factors so that the blastocyst doesn't get destroyed by the woman's immune system, and they're going to form the placenta eventually. Okay, but before that they'll form something called the chorion. All right. The inner cell mass is going to become the embryonic disc and the extraembryonic membranes. And you can see a diagram here of the process, right? So here we have the zygote, right? Then we have cleavage, 2 cells, 4 cells, 8 cells, and then we have the morula, right? Which is a solid ball of blastomeres. It's about 3 days after fertilization. And then it's the blastocyst, right? There's the inner cell mass. There is the outer trophoblast. And then the zona will eventually vanish, and a process called hatching will take place. And the blastocyst will now have just the trophoblastic cells and the inner cell mass and implant into the endometrium, with the inner cell mass being on the side that's closest to the endometrial wall. Okay? And just in these few processes, what you've seen here are all the different processes that lead to human development, right? We've seen cell division, we've seen cell growth, we've seen movement of cells. That's how we got the inner cell mass. Okay, and even though it doesn't look like it, we've had differentiation. The cell mass has different properties than the trophoblast does. Okay, they're not visible to the naked eye at this point, but they're going to go down different developmental pathways. Okay, so all of these things have already occurred. And what guides this are chemical cues that are in the environment that are created by pre-existing conditions. Okay, even at the first cell division, one of the things that we've noticed when we study embryonic development is that the first 2 cells that form are not identical to each other in terms of the composition of the cytoplasm, because they have different distributions of RNA in them, and that RNA was generated while the oocyte was still in the follicle. Okay, and as a result, those 2 cells already have the potential to go down different developmental pathways. Now, a cell will go from a totipotent to a pluripotent to a differentiated cell in the course of development, right? A totipotent cell can become any type of tissue in the adult. A pluripotent cell has multiple fates ahead of it, and a differentiated cell has reached its final developmental destination. Okay, and all this results from the genetic material becoming more and more restricted in terms of what that cell is allowed to use in order to guide what it looks like, what it creates, and what it does. Okay, the other genetic material is still there, but it's not available for transcription. For about 3 days, the blastocyst will float in the uterus, feeding off the uterine milk, which is made by the uterine glands. Implantation is about a week after ovulation. The trophoblast cells are going to adhere to the endometrium, utilizing receptors on their surface. An inflammatory response occurs in the endometrium, and uterine blood vessels become more permeable and leaky, and inflammatory cells are going to invade the region. So you can see here the attachment, right? So there's trophoblast out there. There's inner cell mass. That's going to be you. This is fluid, right? This is endometrium, right? So we're going to begin to burrow, right? The trophoblasts divide and form the cytotrophoblast and the syncytiotrophoblast. The cytotrophoblast gets its name from the fact that distinguishable individual cells make up its composition, and the cytotrophoblast basically forms the outer layer that surrounds the fluid and the inner cell mass, while the syncytiotrophoblast is a multinucleate cell that's going to drill into the endometrium and melt blood vessels and form pools of blood, and eventually form the chorion, and then eventually the placenta and the umbilical cord. Okay, and the term syncytia just means a multinucleate mass. Okay, it's just a term that we use to indicate that in this single block of cell membrane you have more than one nucleus, and that comes about as a result of the fusion of individual cells. Okay, the blastocyst burrows into the lining surrounded by leaked blood. Endometrial cells cover and seal off the blastocyst, and you can see that process here. Okay, the syncytiotrophoblast is undergoing the process of fusion of these individual cells, and it's drilling into the endometrium. The cytotrophoblast remains relatively intact, and there, of course, is you, the inner cell mass. Okay, implantation is completed by the 12th day after ovulation. Menstruation is prevented by the fact that the corpus luteum is maintained in the ovary by the production by the chorion of human chorionic gonadotrophin, which is the pregnancy hormone. Okay, so what this does is it maintains the corpus luteum for the first trimester, while the corpus luteum kicks out massive amounts of progesterone and some estrogen in order to maintain the uterine lining. Okay, so that it isn't shed while we have an implant in there. Okay, after the first trimester, we don't really need human chorionic gonadotrophin anymore because the placenta takes over the production of estrogen and progesterone at that point, and the corpus luteum will then devolve into the corpus albicans, which is not capable of secreting hormone. Okay, so you can see here the syncytiotrophoblast and the cytotrophoblast, and you can see the inner cell mass as well, right? And it's all burrowed in endometrium. Okay, this is a micrograph showing you those different structures. Okay, now during pregnancy, there are a variety of hormone changes that take place in order to support the growing embryo and fetus, and then eventually to initiate childbirth. Human chorionic gonadotrophin, which is made by the trophoblast cells, is going to maintain the corpus luteum for the first trimester, while it in turn cranks out large amounts of progesterone and some estrogen. This promotes placental development through its autocrine growth factor activity, in addition to inducing the luteum to make its hormones. These levels will rise until the end of the second month and decline as the placenta begins to secrete progesterone and estrogen. Low values are going to be seen between 4 months and the remainder of the pregnancy. So you can see how this tracks, right? Ovulation and fertilization, human chorionic gonadotrophin, and the maintenance of the corpus luteum for the first trimester, right? So what you're going to see here is progesterone production after ovulation for the first 3 of 9 months, okay? So 3 months is going to be approximately 12 weeks, okay? So you can see the progesterone levels beginning to peak here, okay? And then after that, the formation of the placenta allows us to increase production of progesterone and estrogen. At this point, the luteum has vanished and has become the albicans, right? Because you can see here the chorionic gonadotrophin levels begin to fall, right? And by this time, the luteum is going to be gone, okay? And that's indicative of the fact that we now have a placenta and we don't need the luteum to crank out estrogen and progesterone any longer. The formation of the placenta is called placentation, and it's a temporary organ that contains both embryonic and maternal tissue. The embryonic tissue are mesodermal cells that come from the inner cell mass lining the trophoblast, and these form the chorion and the chorionic villi, okay? The cores of the villi are invaded by blood vessels, and they extend to the embryo and become the umbilical artery and vein eventually. As they develop in the endometrium, they generate these blood-filled chambers, lacunae, in the stratum functionalis, which are going to bathe these spaces. The intervillous spaces are immersed in blood. The maternal portion of the placenta, called the deciduum basalis, is made up of the functional layer of the endometrium, and the fetal portion of the placenta is made up of the chorionic villi. So you can see here the implantation taking place, the syncytial trophoblast drilling down into the endometrium and inducing the formation of these blood-filled regions from the maternal blood vessels, right? And then you can see these pools of blood beginning to form, and eventually what we're going to have is an interdigitating blood supply made up of capillaries that come from the maternal blood supply lying in very close contact with capillaries that are going to become the embryonic blood supply, okay? So all this out here is chorion, okay? This is yolk sac, this is amnion, right? And this is the trilaminate embryonic disc. This is what is eventually going to be used. So this is endoderm, this is ectoderm, I'm sorry, this is mesoderm, this is endoderm, and this is ectoderm. So the endoderm is going to form the lining of the digestive, urinary, and reproductive systems, as well as the respiratory system. Those parts of the lining that are simple epithelial layers. The ectoderm is going to form the epidermis of the skin and the nervous system, and the mesoderm forms everything else. Okay? So you can see again another example of morphogenetic movement and differentiation, right? We start out simply having the amniotic cavity and the yolk sac over the bilaminate embryonic disc, and then we form the ectoderm and the hypoblast. The endoderm forms next, and the mesoderm forms after that. Okay. The decidua capsularis is the part of the endometrium at the uterine cavity face of the implanted embryo, and the placenta is fully formed by the end of the first trimester. And it helps with nutritive, respiratory, excretory, and endocrine function. So what happens as the placenta becomes more prominent is that the yolk sac begins to shrink and vanish. Okay, the yolk sac is a remnant of when we, back in evolution, used to lay eggs. Okay, and the nutrition for the entire developmental process had to be contained just in the yolk. That's why a chicken egg has a huge yolk sac. Okay, But our yolk sac is relatively small and is meant simply to bridge the gap between um, the formation of the blastocyst and the implantation of the blastocyst and the emergence of the chorion and then eventually the placenta. Okay, the placenta also secretes human placental lactogen and human chorionic thyrotropin and relaxin. So what do these do? Placental lactogen is going to promote the production of milk for the baby. Human chorionic thyrotropin is going to promote the production of thyroid hormone to elevate the metabolic level. And relaxin is designed to soften the, uh, the cartilage between the pubic symphysis, but it also affects the other amphiarthrotic joints as well. Okay? And that's in preparation for the baby's head coming through the birth canal. So you can see here at 4.5 weeks, all of the extraembryonic membranes, right? There's the yolk sac, all right, going in here. This will eventually, all of this will eventually become the umbilical cord, okay? Um, Out on the periphery, right, we have the chorion. There's chorion out there. And then beneath that, we have the amniotic sac with the amniotic fluid. That is going to be your head. This is going to be your body. And this is going to be your legs, okay? These are your arm buds. These are your leg buds. Okay, this is going to be your eye. This will eventually be your face, okay? Um, so the idea here is that this is the first place where blood is actually formed. So we actually um, form the first blood cells um, before we have marrow, okay? And the heart actually begins beating before it has blood to pump, which is kind of interesting in the embryo, okay? Well, let's say as we transition from the embryo to the fetus, all right? But at this point, you can see the chorionic villi with the capillaries that are coming from the the umbilical blood vessels. These would be the umbilical veins here, okay? And then the maternal blood supply, which is still present in pools of blood, but will eventually form um, capillary networks similar to what we have on the embryonic side, okay? So during development, what you have to realize is that the mother's blood and the baby's blood never mix. Okay, that only happens at birth when the placenta is torn from the endometrium of the uterus. Okay, but there is exchange going on. Okay, and that exchange is with nutrients and waste products and oxygen and CO2. Um, Vitamins and minerals get through there as well. Okay, so the idea is what mother eats is what baby also consumes, and this supports the growth and the maintenance of both organisms. Okay, you can see here the placenta. This is 13 weeks, right? And then you've got the decidua basalis here, and the capsularis is above it, facing the embryo. Amniotic sac is larger now because baby is bigger, and then there's chorion outside that. Okay, you can still see the chorionic villi and the chorion on the outer area. Okay, the amniotic fluid is designed to let the baby move and to prevent adhesions, and it eventually becomes filled with the fetal urine. Okay, but it originally begins as a filtrate of blood, right? And you can see how much smaller the yolk sac has become at this point as well. Okay, the allantois is also present, and that's going to be essentially formed in the region here where the yolk sac joins the embryo, okay, and both of these are going to become part of the urogenital system. Okay? If placental hormones are inadequate, the pregnancy aborts. Throughout the pregnancy, the blood vessels are going to contain levels of estrogen and progesterone that continue to go up, and this will result in the preparation of the mammary glands for lactation. We talked about the fact that the embryonic and maternal blood supplies do not mix. Embryonic placental barriers include the membranes of the chorionic villi and the endothelia of the embryonic capillaries. And this is, again, to protect baby from pathogens. Okay. Now that said, viruses can get through, as well as some of the antibodies that the mother makes. So this is one of the times when passive immunity becomes important. Mom's antibodies protect developing baby from pathogens. Okay. But it's critical, especially in the first trimester, that mothers stay away from alcohol and recreational drugs and people who are sick, okay? Because the viruses are mutagens and can cross the placental barrier and can result in birth defects or even abortion. You can see here, as we move forward in the developmental sequence, how now the maternal arteries and veins have become honest-to-God capillaries, and we do have these spaces in here, these sinuses, right, in lacunae that receive the blood from the maternal artery and the veins that drain it, right? But again, notice that the blood supplies do not mix. They just come into very close contact, right? It's all a matter of active and passive transport that determines what gets into baby and what doesn't. Now, another caveat here is that lipid-soluble substances can cross this barrier at will, okay? And so anything lipid-soluble is going to go right through the placenta and into baby and can affect its development, okay? The germ layers are formed in a process known as gastrulation. Right? The inner cell mass becomes an embryonic disc that has an epiblast and a hypoblast. The epiblast is going to face the amniotic cavity, and the hypoblast is going to face the yolk sac. The three primary germ layers are going to form, and the extraembryonic membranes will develop. Right? The amnion is epiblast cells that fill with fluid and provide buoyancy for the embryo and the ability for it to move. It maintains a nice temperature for the growing embryo and fetus. It also is manufactured from maternal blood, but later will contain fetal urine. Okay, the yolk sac hangs from the ventral embryo and will form part of the alimentary canal. Okay, and is the source of the earliest blood cells and blood vessels. Okay, so we transition from yolk sac to blood cells and blood vessels. Okay, but the heart is formed even before all of that. So interesting that the heart begins to beat before it has blood to pump. The allantois is an outpouching at the caudal yolk sac, so it's real close to the embryo, and it will serve as the foundation for the umbilical cord, which attaches to what will eventually be your navel. Right? Portions of it become part of the urinary bladder. The chorion is going to contribute to the placenta and also enclose the embryonic body and all the other membranes. So it's the outermost embryonic membrane. So let's take a look at the embryonic membranes in a little more detail.
During the second week of development, the inner cell mass will flatten to become an embryonic disc composed of several layers. The hypoblast, columnar cells, which will eventually compose the endoderm and yolk sac, and cuboidal cells of the epiblast, which will eventually compose ectoderm and the amnion. The cells of the trophoblast become the extraembryonic membrane known as the chorion. These are embryonic cells, but they will not make up part of the embryo's body. They are an extraembryonic membrane. These will form the fetal part of the placenta, which exchanges nutrients with maternal blood. Also, the chorion secretes HCG, the human chorionic gonadotropin, and the chorion will suppress the mother's immune system so that the mother's immune system does not attack the embryo. Around day 8, cells of the hypoblast contribute to the formation of the extraembryonic membrane known as the yolk sac. Invertebrates which develop in eggs, obviously the yolk is extremely important since it is the source of the nutrition which will sustain the embryo. In live-bearing mammals and humans, the yolk sac does not perform this function, although it does serve for the formation of the first blood cells, the formation of the germ cells, which will ultimately migrate to the gonads for reproduction, and a few other minor functions before it degenerates. 8 days after fertilization, the cells of the epiblast contribute to the formation of an extraembryonic membrane known as the amnion. The amniotic sac will eventually surround the entire embryo and fetus and be filled with an amniotic fluid. This fluid originates as a filtrate from maternal blood, but later urine secreted by the fetus will contribute as well. This amniotic fluid prevents the fetus from drying out, prevents tissues from adhering to fetal cells, and will help absorb shock. And it was the development of the amnion which helped the mammals, birds, and reptiles, the group known as the amniotes, to reproduce on land rather than in water as amphibians do. Beginning 12 days after development, a space develops between the chorion and the amnion, yolk sac, and embryo known as the extraembryonic coelom.
So you can see here again, back at our sequence of events from implantation to the formation of the membranes, right? Same figure that we had before. And there we are again at 4.5 weeks, okay? Showing you all the involved structures. Notice at this point that you have some primitive structures here, right? You have flippers, you have gills, and you have a tail, okay? The only reason I point this out is because there is a recurring theme in development in which primitive structures get replaced by more advanced ones as development proceeds, and biologists have a term for that. Ontogeny recapitulates phylogeny, which is just a fancy way of saying that the events of evolution seem to be like the events of development. So in a sense, we're replaying evolution in miniature every time we generate a new organism. Okay, so you're seeing the whole process play out inside the womb. Okay, very interesting. Gastrulation is at the end of the first trimester. The embryonic disc becomes the three-layered embryo with the ectoderm, mesoderm, and endoderm present. This begins with the primitive streak, which is a raised dorsal groove, and this establishes the longitudinal axis of the embryo. So basically where the head's going to be, where the tail is going to be. It also establishes right and left, okay? So this is on the embryo's backside, right? Cells migrate into the groove. The first population of cells forms endoderm, and then the next population of cells forms mesoderm. So the endoderm and the mesoderm cells have different fates. So they've already been programmed to do different things by the time we're forming these germ layers. And the fact that we're forming these germ layers, these cells are moving through the primitive streak and then layering under the ectoderm, indicates that we're undergoing morphogenetic movement, so we're following chemical cues as we move from place to place. In addition to that, while that's going on, we're undergoing cell division and growth, and we're also undergoing differentiation, right? So all of these processes are happening pretty much at the same time. The next thing to appear is the notochord, which is a rod of mesoderm that serves as an axial support, but more importantly, it's going to induce the formation of the neural tube, which will eventually form the brain, the brainstem, and the spinal cord. So what happens is that chemicals from the notochord diffuse up and then induce in the ectoderm a cavitation over where the primitive streak was, basically, and that results in a crevice along the back of the embryo that will form first the spinal cord, and then the brainstem, and finally the brain. Okay, and again, what we see in that is a progression from primitive to a more advanced structure. The brain was really the last thing to show up in evolution. Okay. Cells that remain on the embryo's dorsal surface are ectoderm. These primitive tissues generate all the different body organs. Epithelial cells and nervous system components come from the ectoderm, right? The endoderm are the simple epithelial linings of the GI, the urogenital, and respiratory, and their associated glands. And mesenchyme forms everything else. So you can see here the progression, right? You've got here the bilayered disc, okay? So you've got ectoderm, and then down here you got hypoblast, right? And then, okay, you have this migration of cells through the primitive streak. This is basically a transverse section through the primitive streak. Here you're looking at a transverse section as well, right? This is looking from the top. The amniotic membrane has been cut away, so you can see from the top, and you can see here the tail and the head, right? And there is your primitive streak, okay? And then under that is yolk sac. So the endodermal cells migrate through and become the new bottom layer, and then the mesodermal cells migrate through and become a middle layer, right? And then that's you. That little disc is you, okay? And then the formation of the neural tube is what's going to follow that, okay? So stay tuned for a look at fertilization and development, okay? Sort of a brief overview of everything we've talked about, and I will join you in the next podcast.
Fertilization is the epic story of a single sperm facing incredible odds to unite with an egg and form a new human life. It is the story of all of us. During sexual intercourse, about 300 million sperm enter the vagina. Soon afterward, millions of them will either flow out of the vagina or die in its acidic environment. However, many survive because of the protective elements provided in the fluid surrounding them. Next, the sperm must pass through the cervix, an opening into the uterus. Usually, it remains tightly closed, but here the cervix is open for a few days while the woman ovulates. The sperm swim through the cervical mucus, which is thinned to a more watery consistency for easier passage. Once inside the cervix, the sperm continues swimming toward the uterus, though millions will die trying to make it through the mucus. Some sperm remain behind, caught in the folds of the cervix, but they may later continue the journey as a backup to the first group. Inside the uterus, muscular uterine contractions assist the sperm on their journey toward the egg. However, resident cells from the woman's immune system, mistaking the sperm for foreign invaders, destroy thousands more. Next, half the sperm head for the empty fallopian tube, while the other half swim toward the tube containing the unfertilized egg. Now, only a few thousand remain. Inside the fallopian tube, tiny cilia push the egg toward the uterus. To continue, the sperm must surge against this motion to reach the egg. Some sperm get trapped in the cilia and die. During this part of the journey, chemicals in the reproductive tract cause the membranes covering the heads of the sperm to change. As a result, the sperm become hyperactive, swimming harder and faster toward their destination. At long last, the sperm reach the egg. Only a few dozen of the original 300 million sperm remain. The egg is covered with a layer of cells called the corona radiata. The sperm must push through this layer to reach the outer layer of the egg, the zona pellucida. When sperm reach the zona pellucida, they attach to specialized sperm receptors on the surface, which triggers their acrosomes to release digestive enzymes. enabling the sperm to burrow into the layer. Inside the zona pellucida is a narrow, fluid-filled space just outside the egg cell membrane. The first sperm to make contact will fertilize the egg. After a perilous journey and against incredible odds, a single sperm attaches to the egg cell membrane. Within a few minutes, their outer membranes fuse and the egg pulls the sperm inside. This event causes changes in the egg membrane that prevent other sperm from attaching to it. Next, the egg releases chemicals that push other sperm away from the egg and create an impenetrable fertilization membrane. As the reaction spreads outward, the zona pellucida hardens, trapping any sperm unlucky enough to be caught inside. Outside the egg, sperm are no longer able to attach to the zona pellucida. Meanwhile, inside the egg, the tightly packed male genetic material spreads out. A new membrane forms around the genetic material, creating the male pronucleus. Inside, the genetic material reforms into 23 chromosomes. The female genetic material, awakened by the fusion of the sperm with the egg, finishes dividing, resulting in the female pronucleus, which also contains 23 chromosomes. As the male and female pronuclei form, spiderweb-like threads, called microtubules, pull them toward each other. The two sets of chromosomes join together, completing the process of fertilization. At this moment, a unique genetic code arises, instantly determining gender, hair color, eye color, and hundreds of other characteristics. This new single cell, the zygote, is the beginning of a new human being. And now the cilia in the fallopian tube gently sweep the zygote toward the uterus, where he or she will implant in the rich uterine lining, growing and maturing for the next 9 months until ready for birth.